Hello, everyone, and welcome to today's live broadcast, Osmolality Testing, Providing Unique Process Control for Downstream Operations, presented by Dr. Alois Jungbauer and Dr. Kristeena Wright. I am Michelle Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Labroots and sponsored by Advanced Instruments. For more information on our sponsor, please visit aicompanies.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenters, Dr. Alois Jungbauer, Osmolality Testing, Providing Unique Process Control for Downstream Operations, and Dr. Kristeena Wright, Application Scientist at Advanced Instruments. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Dr. Jungbauer and Dr. Wright, you may now begin your presentation.
Thank you so much for that introduction. My name is Kristeena Wright, and today I will be talking a little bit about osmolality testing, and we'll be joined, as was mentioned, by Dr. Alois Jungbauer of the BOKU to talk about how this test really provides unique process control for downstream operations. Just to give you a little bit of insight into what we'll be discussing today, we do want to start at the beginning, really talk about what osmolality is as a test, how it applies to bioprocessing, where it really fits in within a bioprocessing workflow, and then we'll get into the good stuff. We'll get into some new data that Dr. Jungbauer would like to present, kind of go over a study that he oversaw and discuss some key takeaways. Then we'll wrap up by discussing how Advanced Instruments has really provided the devices and the technology that's necessary to implement osmolality testing, no matter where you are in your workflow, but particularly for downstream processing. So let's go ahead and get started. If we want to start by just defining osmolality, it's a colligative property of any liquid solution. So anytime a solute is introduced into a solvent, the resulting solution is going to differ from that initial solvent in several ways, and osmolality is one key measurement to really pick up on those differences. It's a concentration measurement. It's the total number of solutes in a liquid solution, typically expressed in milliosmoles of the solute per kilogram of the solvent. And that's important. We'll get a little bit more into why that measure of mass of the solvent is so important in a second. Because osmolality is not dependent on volume, but on the mass of the solvent, it's not necessarily affected by a change in the solution's temperature. So you could start to see why this may play an important role when it comes to bioprocessing, where temperature, pressure, different parameters are changing. One question I know that I get a lot when we talk about osmolality testing is, what exactly is the difference between osmolarity with an R and osmolality, which is what we're talking about today? So I wanted to dive a little bit deeper into this and give some really clear insight into what the differences are. So I mentioned that osmolality is a concentration expressed as osmoles of solute per kilogram of solvent. This is different than when we talk about osmolarity, which is actually osmoles of solute per liter of the total solution. And you can see in that second point, while osmolarity is actually dependent on temperature and pressure of the solvent, because volume itself is temperature dependent, osmolality is not something that's dependent on the temperature and the pressure of the solvent that you're looking at. So what we tend to see is that osmolality is actually a preferred concentration measurement within biological systems. And this is something that really plays a role into the study that Dr. Jungbauer will be discussing later. So I'd kind of like to bring you in right now, Dr. Jungbauer, and ask, where is this temperature independence? When we talk about osmolality testing, where is that most relevant in your experience?
I think it is extremely relevant when you prepare buffers, mainly buffers with through a stock solution in a high solute concentration. And there are a couple of unit operations in bioprocessing or downstream processing which are really sensitive to temperature. One example, for instance, is hydrophobic interaction chromatography or reversed phase chromatography. So these two unit operations are extremely important, extremely dependent on temperature. Yeah? So the retention time or the yield would highly depend on the temperature. So when you actually, for instance, prepare a buffer at 4 degrees and then use the buffer at room temperature, pH and conductivity would change. Osmolality does not. I think this is one of the prime examples.
Perfect. Okay. And that's a really great point. So I think it sets us up really nicely to get into more of the study that you were able to do and recently published on. So we'll get into more of that data in a bit. One thing that I did want to kind of go through, because obviously today we want to focus on purification, but we also wanted to work through and kind of discuss some of the applications for osmolality testing in other areas of bioprocessing. And of course, feel free to reach out. We do have materials and extra resources available that kind of speak to some of these applications as well. So if we start with more upstream monoclonal antibody processing, let's just take kind of an overview of where osmolality testing would fit into cell line development. So really when we want to ensure that the cell media and culture conditions are optimized so that clonal screening and selection and characterization can be done efficiently and effectively, osmolality testing is a really great parameter to put into place at this time. Setting a range for osmolality really sets you up for productive and stable cell lines so that you can get further processing done more efficiently. With media preparation and quality control around media preparation, it's important to check the osmolality of any raw materials that could be going into the media. And then the final media composition or concentration itself should definitely be measured using osmolality testing, just to make sure that the cultures are really able to thrive. You want to have that optimal cell viability and ultimately yield. So osmolality is definitely a parameter here that should be maintained and checked. And during the process of cell culture monitoring, whether you're working with shake flasks or bags or bioreactors, osmolality is something that should definitely be monitored and considered a critical parameter as this process is ongoing to ensure that all the conditions are really optimized for ideal growth, for ideal health. If there are other parameters that you need to keep in check, this is a great way, a very robust measurement to make sure that that's happening as far as the media and the concentration of the media is concerned. So we spoke to you a little bit about downstream processing. Alois spoke to some of the applications with buffer preparation. Again, similar to with media preparation, making sure that you know exactly what's going on. This is a really credible and robust measurement to make sure that you understand the composition and concentration of your buffers. As you work through the key operations, whether it's chromatography or filtration, today we'll be focusing more on the purification, the chromatography steps. So again, this is where we'll speak a little bit more to how osmolality testing can be used to predict protein quantity and potentially identify any process deviations. But there's also certainly an application within buffer exchange or really any filtration operation. But when we talk about buffer exchange and making sure that you can, if you already have a set number of exchanges or diavolumes, tracking that concentration as you're going through your exchanges, or maybe even optimizing and reducing the number of exchanges used. And when it comes to the buffer preparation and QC, and just speaking to these buffers that are used throughout downstream processing, it's critical, as Dr. Jungbauer mentioned. So we did want to show some data from a study that we completed last year. And while simple, it really does show some pretty key information when it comes to dilution curves. And as the concentration of key buffers and buffer components change, you tend to see, while maybe not perfectly linear, you tend to see more of a traceable, predictable pattern with osmolality over those concentrations that really take up the full span and possible range of osmolality testing. If we compare that to something like conductivity, certainly we're not seeing a linear curve, but another issue is that, for example, with something that's weakly dissociating, like acetic acid here, you tend to see that at really high concentrations, there's not that resolution. There isn't a great distance between the conductivity of this acetic acid at, let's say, a concentration of 1 molar versus a concentration of 2 molar. And while we know that that teams are not necessarily working at these higher concentrations throughout the downstream process, we also are seeing a trend in inline dilution systems, for example. So if you start with these high concentrated stocks, then you will have to kind of consider what the concentrations of those starting materials are. So that's why we wanted to take in a full range of the concentration of these buffers. And last but not least, if we talk about formulation and the role of osmolality testing in formulation and final product testing, for formulation development, certainly screening potential formulation concentrations with osmolality could be key, especially when you have a lot of different conditions and formulations that are being tested. We certainly offer ways that you could do this more high throughput and multiple tests in a run. When it comes to final formulations and verifying concentrations of excipients, this can also be achieved with osmolality testing. Additionally, osmolality has been associated with the injection site pain experience with some of the final formulations and the final products of MABs. And lastly, osmolality is actually a compendial test, so it's something that we pretty much see 100% use for that last identity measurement and specification. So, of course, this is an area where we see a lot of value for this test. So that's just a little bit of an overview at each of these areas. Of course, we are more than happy to provide additional information, but we at least wanted to present the general idea of where we fit when it comes to upstream, downstream, formulation, and final product. So now we will actually get into the exciting data that Dr. Jungbauer recently published and kind of speak to more of a novel area, really, of osmolality testing when it comes to being a predictor of protein concentration during chromatography operations. So Dr. Jungbauer, if you want to take over, I will give the floor to you.
Thank you, Kristeena, for this introduction to osmolality and actually explaining the difference between osmolarity and osmolality. Actually, there is quite a big confusion in the scientific community about these terms, or maybe for a lot of people, osmolality actually is a new term and they have not heard before. The work we have actually done in this context is done within a bigger frame. We are interested in real-time release. Yeah? And this is actually the testing of our process and in-process data, yeah, to actually know the quality of the product at any stage in the process. And then after you have finished the process, you are aware, or you know that your product is within specification. And therefore, we measure a combination of material, attributes, and process controls. And actually, there is a guideline for a couple of years already available from FDA and the International Committee of Harmonization, the ICH Q8 guideline on real-time release. Yeah? We should be aware that real-time release actually helps us to improve product quality, the consistency, but also adds on a lot to economics. Because we assume, or it is assumed, that with real-time release, batch failures can be substantially cut down. The bottleneck for the real-time control and real-time release is actually often the product-specific inline sensors or fast online monitors. And actually, these fast online methods can be replaced by statistical models, but this is also known under soft sensors. So actually, I will explain you in a couple of minutes about real-time monitoring and statistical modeling of downstream processing. On the right-hand side, you see the reference where we have published this data, and you find more actually information and also the references. I think I advise you could also check our annual report of the Institute of Bioprocess Science and Engineering, where you will also get more information. So what you do actually, you connect your chromatography workstation to different sensors, and do online and at-line measurements. So you would have offline signals and online signals. And then with these online and offline signals, you do a statistical model. That means the offline signals are used actually to predict the online signals, and then you get an idea of your process stream in real time. So what we have shown in this publication, that we are able to predict host cell protein, DNA, host cell DNA, and some product quality in real time. So now, actually, together with the company Advanced Instruments, we wanted to explore a bit the effect of osmolality as a predictor for our real-time monitoring and statistical modeling. So, we did a flow-through chromatography, like a polishing step to remove aggregates from monomers in an antibody process. Below you find the paper which we have published recently, and there you find all the details. I just want to explain you one actually simple statistical term. This is the RMSE. This is the relative mean square error. This is actually the sum of all errors between the measured and the predicted values. Yeah? So when we assume that we take osmolarity into account, then we see that with osmolarity, actually we get a lower, or this RSME, a lower prediction error compared to UV 280 to 60 to 14 pH and conductivity. So if you look the experiments, I don't want to go to all the experiments we have done. Yeah, on the lower right corner, of course, osmolarity alone would not be able to predict actually this product quantity. But UV 280 to 60 and osmolarity have actually a substantial lower prediction error. And this was also puzzling us when we wrote up the manuscript, because you need an explanation why actually is the osmolarity alone better than pH and conductivity together with the spectroscopic methods. I think one reason is actually what we capture with the osmolarity is actually the number of solutes, and what we also have mentioned in the paper is actually the osmolarity itself. Yeah, captures much faster actually all the transition in the composition of the process fluid. So you see here, I already mentioned, we published this this year in the Journal of Chemical Technology and Biotechnology. And actually, if you want to get more information, I advise you to read this paper. So that for us was really actually a surprise that osmolarity is a really good predictor. Also, we should have known it in advance because the osmolarity itself, yeah, is highly dependent actually on the change of solution. You know, the buffer itself, yeah, changes actually with the buffer pH changes with dilution. Yeah, the conductivity concentration relationship is not fully linear. This has been already elaborated by Kristeena. And I think if we go back to our basic chemistry, we should consider Ostwald's dilution law and therefore including osmolarity, because we know that the dissociation of constant of a weak acid or base actually is highly dependent on the concentration. So now we have to ask ourselves, how could be this also translated into newer actually areas in bioprocessing, in cell and gene therapy? Because it is well known in the field of formulation, yeah, that the osmotic pressure has a high influence on the viability of the cells. On the right-hand side, you see this prime example, the red blood cells. In the middle, you see how happy the red blood cells are. They look like donuts in isotonic solution. Actually, in hypertonic solution, actually they start to actually increase in volume until they would burst. In hypertonic solution, they shrink, yeah, and actually get the kind of a blur. So, therefore, it is really important in this field to keep our bioprocessing solutions, also during downstream processing, the cells happy in isotonic conditions. So that means when we prepare buffers, loading buffers, wash buffers, elution buffers, actually, it's really important to actually check our osmolality, because this will increase yield, improve the quality of cells or viruses, yeah. And in case of stem cells, it's known that it would remove biophysical threats from cells, yeah, which could lead to an apparent dissociation. So this was a kind of outlook into the future, where also osmolality would be very important. And now I want to actually hand over again to Kristeena.
Thank you so much, Dr. Jungbauer, and I definitely appreciate you speaking to kind of how this would also apply and translate to the cell and gene therapy space. I know that that's a question that we've gotten before, and it's certainly an area of interest for a lot of people in our field. So we look forward to kind of working our way into that space and hopefully getting some really valuable insight from you and your team, even when it comes to the cell and gene therapy space as well in osmolality testing. Thank you so much for that report. We're certainly very excited about the data and kind of the story that comes from that, and also to see, you know, where it leads us. Like I said, whether it's into cell and gene therapy or advancements with purification processing. So one thing that I did want to provide is we've talked a lot about osmolality testing, but the question is, how do we accomplish this? And what is Advanced Instruments doing to make sure that this is something that is accurate? It's a test that's robust, and really, as far as what services and what offers we can provide, we want to make it something that's really easy for our customers to obtain and then to use. So just to give you an idea of what types of instruments we have available, I did want to show we have a single sample microosmometer as well as a multi-sample microosmometer that are now available. That would be the OsmoTECH and the OsmoTECH Pro, respectively. So the major difference here is with the OsmoTECH Pro, you can run up to 20 samples in one sitting. That will kind of automatically run around that carousel as opposed to running one at a time. So really it has more to do with the throughput and however the number of samples that you need to run in one sitting or in one day. And we can certainly help you figure out what the best instrument is for you. However, they both offer really a lot of great features and benefits for those in biopharma and biotech. As you can see, there's a lot of those features listed here. The accuracy and precision that Advanced Instruments has really become known for. Of course, that's something that we've carried through to this OsmoTECH portfolio. Really great settings and permissions that provide flexibility. So we recognize that there are customers that really have to focus in on things like 21 CFR Part 11 and that data compliance and that data security. Whatever you need when it comes to that, we have with these instruments. However, if you're at the other end of the spectrum, you don't need that right now, or it's not something that you need maybe fully installed, there is that flexibility to work within different parameters, and that's really just up to the user for each of these instruments. Also, as far as testing time, while this would be an at-line measurement for those of you that are in purification, it's a 90-second test time for each test, for each sample run. So it's relatively fast, but still providing you that accuracy and precision that you need. So the other thing that's really unique about our instruments is the data management. And you can see here that we have several ways now that you can get data very quickly from the instrument into your system, into your database, whatever it is, whatever kind of electronic records you have or systems that you use, from OPC to an embedded web server where you can view the data remotely. We have the LIS and the LIMS connections available, as well as the ability to push the data from the instrument into your network pretty seamlessly. You can do that automatically. You can set it up to do it at a pretty regular cadence, or it's something that you can do manually whenever you need it. So you can see all of those different ways are listed here. And again, just allowing flexibility. So if it's something where you're looking to upgrade to a better level of connectivity, you can. If you're already printing out results and you want to keep it that way for now, but you want the option to kind of move ahead in the future, we do offer that with the OsmoTECH line of osmometers. And the service and support, I mentioned this, but just wanted to bring your attention to this as well, because it's not always just about the instrument, but also making sure that the installation is there. We offer training. Validation services are also provided along with each of these instruments if that's something that you wish to have. Preventative maintenance, and of course the technical support that you would need to make sure that if you do run into any issues with your instrument, they can be resolved quickly and thoroughly. So really, just to kind of wrap up everything that we've discussed today, osmolality testing is definitely a valuable orthogonal tool as far as measuring buffer concentration within downstream workflows. So this means more than just that buffer QC measurement. And as Dr. Jungbauer explained, because it's independent of temperature, it's something that really becomes a valuable tool within bioprocessing. And when we speak to purification in particular, you're measuring those buffers throughout this process. It's an in-process test, not necessarily just a check it and leave it type of test. So the new publication specifically, and again, we are happy to point you in the direction of the paper from Dr. Jungbauer's team if you'd like more information on that. But it shows that osmolality can serve as an in-process indicator of protein quantity, and also potentially, as a result of that, indicate any deviations. So as he said, and as he showed in the graphs, osmolality, along with UV 260 and 280, can be used as inputs for a very simple statistical model to predict the protein concentration for your MABs. And although osmolality is not yet available as an online measurement, Advanced Instruments does offer osmometers that can provide really simple, accurate, and fast at-line testing. They come ready to fit into your workflow, so no matter what your needs are, again, as far as data integrity and connectivity, we can meet those needs and definitely work with you to get everything set up in an efficient way. With that, we'd just like to thank you so much for your attention today. And on behalf of Dr. Alois Jungbauer, I'd like to invite you at this time to ask any questions. Thank you so much.
Thank you, Dr. Jungbauer and Dr. Wright, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's go ahead and get started. Okay, our first question is, with respect to upstream cell culture process for monoclonal antibodies production, what ranges of osmolality are appropriate for cell viability, growth and for monoclonal antibodies productivity?
I think there is a pretty actually narrow range, of osmolality. I think this is very well documented in literature. I think also Advanced Instruments has application notes about this, because at higher or lower osmolality, that's in the range of 400 milliosmol, you might actually change the viability of the cells, and this puts on stress on the cells. But I think there are cells around which are more resistant or to high or low osmotic pressure than others. Maybe also, Kristeena, you could comment on this,
Sure. I totally agree. And I would say, of course, it's going to depend on what cells you're working with. Most of our customers are typically, if they're working with CHO cells, reporting something around the 290 to 300 milliosmol per kilogram of water range. However, we have definitely, like you said, Dr. Jungbauer, we've seen a range, and it depends on the cell line. It depends on what your goals are from your cell culture. So we're happy to kind of work through that with you. If it's something that you're interested in, please feel free to contact us.
Great, thank you. Okay, let's move on to our next question. What osmolality range helps in clearing monoclonal antibodies aggregates in the purification process, and at what step?
So should I answer this question, or Kristeena should go ahead? I say hello. Yeah.
Sure.
You can hear me?
You can go ahead. Yes.
Actually, usually you remove the aggregates either in the intermediate purification. Nowadays, the flow-through chromatography is quite popular for this purpose. That because it's known that, for instance, the aggregates bind stronger to, for instance, cation exchanger compared to the monomers. So, but usually this process is very sensitive to a salt concentration. So you would go actually to conditions where you just actually pass through the actually monomers, and you bind the aggregates. So this is actually a very subtle process and needs a lot of fine-tuning. Another more robust process is actually hydrophobic interaction chromatography for removal of monomer aggregates from monomers. Because here it's the same. The aggregates bind much stronger compared to the monomer. And so the monomer will be eluted actually later, earlier compared actually to the aggregate.
Thank you. How would this model apply to capture chromatography operation rather than flow through?
What do you mean? The method, the osmolarity measurement, I did not completely understand the question.
So I think if your setup involved rather than a flow-through process, capture
I
process.
think if you want a flow through or a bind elute process, I think
Right.
this really depends also on the company, on the history. What I mentioned before, maybe it's more difficult in a flow through operation to find robust conditions compared to a bind elute process.
Great, thank you. Okay, our next question is, if we want to try this modeling method alongside purification, what is the best way to get started?
So what is meant with the best way to get started if we do like a modeling or so? Yeah? For the modeling, actually, if you really want to go into this real-time release things, is actually you have to actually set up either a couple of training runs, or I think quite often is available. You have already repetitive runs in your company. And then this would be great, yeah, because you have already the UV data and so on, and you could try actually to establish a simple mathematical model. And later on, you can actually include more and more online and at-line sensors. Yeah. And actually what we have done, and was the surprise that the osmolality, we said as an at-line sensor, which is actually works quite fast, gave us very promising results.
And I think too, thank you for that. And also I think to add on to that, if you're interested or need any support from Advanced Instruments, obviously we have a team that can respond to whatever needs you have. And Dr. Jungbauer has been great as far as kind of advising on this model, obviously. So we're more than willing to help you get started and make sure that you have, the equipment and understand kind of the process to get all of that started so that it's nice and smooth and efficient for you.
Great, thanks so much. Okay, next question is, what is the difference between upstream and downstream osmolality during monoclonal antibody production?
Kristeena, do you want to answer this question, or should I answer?
We could both. I think so. I'm not sure if the question is specifically about the range, but maybe I think some of what you presented earlier, Dr. Jungbauer, sort of spoke to that. So if you want to start, and then I can definitely kind of add if there's anything else.
No, I think in the upstream processing, it is well established that the people actually use actually a medium which is well balanced regarding the osmolality or osmolarity. Yeah, so that's actually this is historically, yeah. Nowadays, actually, quite often you can actually get more insight, yeah, when you develop a process for the cultivation, when you have actually methods at hand where you can easily measure the osmolality, yeah. Because if your actually upstream processing is not working properly, yeah, you would actually get a hypertonic solution. at the very end, because maybe you actually titrate your bioreactor, the broth, the cells with actually a base, for instance, and then you add more and more solutes, and the osmolality increases. In downstream processing, honestly, except the formulation development, osmolality has not been used so far. Yeah, so this is a completely new area.
Right, and I think to your point earlier, also the focus maybe shifts, right, from
the focus maybe, yes.
From the health of the cell, and trying to make sure that you have productive cells, you have that great yield coming out of your upstream process, and then in the downstream, as we showed you, just kind of looking at your buffers, which are now interacting with your drug substance and the proteins. It's a little bit, the focus kind of shifts on where you may be looking at osmolality.
I think where it's also very important to use the osmolality testing, or very convenient, because it's temperature independent. What we mentioned at the beginning is with this inline dilution, or the dilution. Right. A lot of companies nowadays, they buy actually stock solutions for their buffers, and then they dilute it. And when you use osmolality as a parameter, then it is much more accurate compared to conductivity or actually pH and pH, because at very high salt concentration, the conductivity measurement may fail. Fail means you get inaccurate, or you have to dilute your stock solution to measure the accurate conductivity. With osmolality, you have a quality parameter because you can measure this stock solution without further dilution.
Great, thanks so much. Okay, now this next question is a two-part question. It says, first off, thank you for your presentation. I am working with recombinant proteins. Final formulation based on A280 or A280 is in the region of 0.3 to 0.5 AU. Can osmolality provide consistent and precise results?
So in the final, I think to measure the osmolality would definitely help you to add or to get a higher, let's say, a more robust process for the final formulation. Yeah.
Now, also, they say
and once you have like a free store and you have some precipitation, definitely you would actually see this, because then quite often the protein precipitates with other solutes and so on. Yeah. Because then you see really the change of the solution properties after freeze thaw.
Great, thank you.
Yeah.
Okay, your next question is, how do you measure osmolality within a cell?
I think maybe currently there are some very sophisticated physical methods around, but it's not possible to measure the osmolality in a single cell, in the cell itself. What you measure usually is the osmolality of the broth. Yeah. So of the medium and not the osmolality in the cell, because the osmolality in the cell and the osmolality in the medium would be different. And this is the reason why you want to have physiological conditions in your cell culture or in your bioreactor, because the cell needs energy to maintain actually the physiological condition within the cell. Yeah? And if the surrounding is actually not physiological, it takes a lot of energy to maintain these conditions inside the cell. Either they have to pump out actually solutes or to pump in water.
Now, is it possible to modulate protein aggregation within a cell using osmolality of the cell media?
I think this is definitely, I'm not really a real expert in upstream processing, but it has been shown that actually the wrong osmotic pressure in a cell culture or in a culture may lead actually to protein aggregation.
Great. Now your next question is, can advanced instruments 3250 single sample osmometer be used for USB, network, or other options to download data?
So currently the 3250, we do not have those same network connectivity options that we offer with the OsmoTECH and the OsmoTECH Pro. But certainly if you currently have a 3250 and have any questions about potentially switching to another instrument or need additional information on our products on the OsmoTECH line of instruments, please feel free to reach out to us, and we can certainly work with you to find a solution if your connectivity needs aren't currently being met.
Great, thank you. That looks like we have time for one more question. And this question says, can you speak to the potential savings of implementing this model in downstream processing?
It's difficult to say the time, the cost savings, yeah, or operational costs. I think what we mentioned before, implementing actually osmolality measurements will be actually you would get more robust processes. And of course, if you have a more robust process, this at the end would actually also lead to cost savings or time savings.
Okay, it looks like we just got one more question in from the audience, so let's go ahead and ask this one before we close. And it says, are there chemicals known to interact with osmolality measurement? I'm working with formulation buffers that have polysorbate in them and was wondering if that would have an impact on the accuracy of the measure.
I think this is the beauty of the osmolarity, that actually it measures the amount of osmolytes of dissolved compounds in the solution, and actually it captures if there are interactions. The osmolarity measurement would capture this. Yeah. So it's the number of dissolved compounds. For instance, if you would add a cationic compound, yeah, into your formulation buffer, and the cationic compound actually binds some anions, yeah, then you would see this when you measure the osmolality.
Yeah, and if I can add, this was actually, I'm glad that we got this question. It's a question that we've gotten before with especially more viscous, either reagents or solutions or whatever it is that's added to different samples. We've gotten the question of whether freezing point depression is an answer, is a solution for osmolality testing. And we certainly, our technology has proven to be pretty robust when it comes to a lot of these samples. If it's something that you're interested in testing out before, potentially making an investment, we do also offer sample testing as well. So please feel free to reach out to us. And also, speaking of tween, we have some previous samples that we've gotten before with different concentrations of things like polysorbate, 20 and 80. So we can certainly field your specific questions, and just feel free to contact us. We can certainly work with you on that as well and figuring out which instrument's the best for you.
Thank you again, Dr. Jungbauer and Dr. Wright. Do you have any final comments for our audience?
I don't think so. I do definitely want to thank everyone that's attended. We certainly appreciate your time and attention and look forward to sharing more data and more great information with you in the future.
Okay, no, I also don't have any further comment. I actually also thank you for the questions, because I also learn through these questions. Thank you.
Thank you. And before we go, I'd like to thank the audience for joining us today and for their questions as well. Questions we did not have time for, and those submitted during the on-demand period, will be addressed by our speakers via the email addresses you provided at the time of registration. We would like to thank Dr. Alois Jungbauer and Dr. Kristeena Wright for their time today and for their important research. We would also like to thank Labroots and our sponsor, Advanced Instruments, for underwriting today's educational webcast. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We hope to see you again soon. Have a great day.